Hello, very good afternoon to the student who have joined. Please wait, we will start shortly. So, very good evening to the students who have joined. Can you please let me know in the chat box if my voice is audible to you? And also, please let me know if you can see the presentation and if you can see the video, my video clearly. In the chat box, please confirm if my voice is audible and if you can see the presentation and also if you can see the video. Okay, thank you for confirming. We will start at 6 p.m. So please wait till then. Meanwhile, let me know if you have any doubt from our previous session. In our previous class, we have discussed various aspects of molecular biology and also we have discussed a little bit about the cofactors and coenzymes. So if you have any doubt or any query regarding those, you can let me know while we are waiting for more students to join. Otherwise, it is fine. We will wait till 6 p.m. and then we will start today's session. Okay. Hello. Can you please let me know if my voice is audible or not? Because there is a power cut issue. Is my voice audible? Okay, okay. Thank you for confirming. Sorry, there might be some issues with the electricity, so please bear with me. There might be some kind of electricity issues today. So if I get disconnected for some reason, please do not leave the call. I will rejoin as soon as possible. So let us wait for 2 more minutes, then let me start today's session. Again, I am saying if you have any doubt, any query, please let me know. And there might be some connection issue, connectivity issue. So please do not leave the session if there is any disturbance. I will try to rejoin as soon as possible. Okay. Okay, so I think I will start the session now. If you have any problem with the audio or video or anything, please kindly let me know. Okay, I hope the slides are visible. If not, you can let me know in through the chat box. So I welcome you in this week 8 live session of the NPTEL SWAYAM course of Organic Chemistry and Biology and Drug Development. A very good evening to all. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAM course of Organic Chemistry in Biology and Drug Development, and today it is our week 8 live session. So every live session, what we do is we take that particular week's course content. Okay, every week's course content is released on on Friday, I think, and after 1 week we have that particular week's we discuss that particular week's course content after 1 week. Okay, so you have entire 1 week to go through all the class materials, and then we will we have this kind of live session where we discuss around 20 MCQ questions. All the questions are from that particular week's course content. Okay, and every question is with four different options. All are MCQ questions and one options are correct. And how we do in every class, like after showing you a particular question, I will wait for some time, and you have to let me know what is the correct answer through the chat box, and I am monitoring the chat box every time. So if you have any doubt or any query, also you can let me know through the chat box. So before starting the session, let me clarify a few things. Okay, so basically there will be this kind of problem solving session every week, and these sessions are scheduled on Friday from 6 to 8 p.m. The sessions are live on Google Meet platform. That, as you can see on the schedule, the sessions are live, and this is the meeting link. You can write it up on some time because this link will be same for our entire course, and we will have this kind of, since it is a 12-week course, so we will have around 12 live sessions for this organic chemistry and biology and drug development course. And after that, I think we will have a extra session. We may have a 1 hour extra session which where I will summarize the topics of what we have discussed till now. Okay, so the important thing, if any kind of network disruption happens, please wait for a few minutes. Do not leave the session. I will try to rejoin as soon as possible. Okay, since I am recording the session, I have muted your microphones and cameras, but the chat box is always open. I think you know, whoever is doing the classes regularly, they know that I communicate through this chat box. So if you have any kind of doubt, query or anything, please let me know through the chat box. I'm monitoring the chat box every now and then. So if you have any problem, anything, you can share through the chat box. I will look into it and I will discuss that particular thing. After class, if your doubt persists, you can drop an email to me. I will answer your queries through email, or if it is not possible to do it by email, I will discuss that particular doubt in the next class. Okay, and this presentation and class recording, it will be shared to you. So where you have to find this presentation and class recording, I have shown you earlier also, but let me show you one more time. You have to go to the SWAM portal. I think you can see my screen, right? So if on the SWAM portal, wherever there is this organic chemistry, biology and drug development, so you will see there are announcements of regular course content, presentation, and around according I mean to the for the feedbacks also, and for course content, and also you are getting reminders for the problem solving sessions here. So here on the left panel, where there is a weekly contents, you will find that after every content there is a quiz which you have to take, because it is marked and there is a particular deadline. For example, today it is 12th of September, so you have deadline for submitting the quiz for week 8 assignment by 17th, I think. So you can go to it and you can check. And also here you will find at the end, you will find the something called problem solving session. If you click on this problem solving session thing, you will find there will be a page. You will be redirected through this. There you will find every classes YouTube link. All the classes are posted on the YouTube, so you can go through the videos to check whatever you have missed. If you cannot join the session, you can go through the materials through the videos. Also, you will find this presentation which I am presenting today, it will be also be shared to you. Okay, this presentation will be also be available in this Google Drive. The link of the drive is also given to you. You can download from here easily. You download the presentations from here easily. Okay, so these are the things which I had to summarize before starting the session. So as I have mentioned, today it is our week 8 live session. So what we have discussed in our previous live session. So in our week 7 live session, we have discussed about the molecular biology. Before that, we have discussed about the central dogma and the topics which we have not discussed at that time, that we have discussed in our previous session. Through the molecular biology, we have discussed about the restriction enzymes. We have discussed about the DNA recombination technology, and we have discussed about PCR and all those things, and little bit we have also discussed about the cofactors and coenzymes. Okay, that thing we had discussed in our previous class. So, what is the course content for this week 8 of this organic chemistry and biology and drug development? So, it is basically the chemistry of cofactors and coenzymes, which we will discuss in today's class in detail. So, whatever we have. So actually enzymes, so what you know is enzymes are the catalysts for the biological various transformations, right? So what the enzymes do, they basically help a particular biological reaction to go on smoothly and to go fast, and by lowering the activation energy, just like the catalysts do for a chemical reaction, right? So I mean about the enzymes and all those and modes of enzyme inhibitions and various kinetics, we have already discussed in our week 3 or 4. I do not remember correctly, but I think in week 2 or 3 we were discussing about all those things, right? So now today we will discuss the thing which is the chemistry of cofactors and coenzymes. So what is a cofactor, what is our coenzymes, and what are the difference and everything that we have discussed already in our last class. You can go through that particular presentation. If you do not remember few things, you can go through and you can just have a look from that. Okay. So like I have mentioned that today also I will show you around 20 MCQ questions. Each question we will have 4 different options. These question solving will help you to understand the course content clearly, and also it will help you to answer in your weekly assignments, right? Because the weekly assignments are given in MCQ format. So that is the goal of the live session, so that you can understand the course content better, and also to make you more efficient in answering the assignments questions, and also for the quizzes, right? So yeah, one more thing, the answering in today's classes questions, there will be no marking, but the assignments are marked. So you have to be careful while answering the quizzes. But today you can answer freely. Even if your answer is wrong, it is fine. Okay. So let me start with the first question of today's session. So the first question of today's session is, what is holoenzyme? Your options are: number A, inactive protein portion of an enzyme; number B, active complete enzyme; number C, non-protein portion of an enzyme; and number D, enzyme without any metal ion. What is holoenzyme? Again, I will repeat the options. Number A, inactive protein portion of an enzyme; number B, active complete enzyme; number C, non-protein portion of an enzyme; and number D, enzyme without any metal ion. What is holoenzyme? If you have gone through the previous classes presentation, I know all of you can answer it clearly, because or very easily, because I have already discussed what is a holoenzyme in our previous class as far I remember. So let me know in the chat box A, B, C or D, which of the following you think is the correct answer for problem number 1. I will wait for 1 or 2 minutes, then I will check the chat box. I want all of you to answer. Even if your answer is wrong, it is fine, but please try to answer the question. What is holoenzyme? Number A, inactive protein portion of an enzyme; number B, active complete enzyme; number C, non-protein portion of an enzyme; and number D, enzyme without any metal ion. Let me wait for some time. So what is holoenzyme? Your options are: number A, inactive protein portion of the enzyme; number B, active complete enzyme; number C, non-protein portion of an enzyme; and number D, enzymes without any metal ion. Let me check the chat box now. I hope to find some answers there. Okay, I got 2 answers. One is saying it is number B, other is saying it is number C. Let me check which of the following is the correct answer: number B, number C, or anything else. Okay, so the correct answer will be, it is number B, active complete enzyme. It is known as the hollow enzyme. Okay, very good. So I have discussed what is apoenzyme in last class, if I have remember. So apoenzyme is the protein portion which is inactive. So every enzyme has a particular protein portion, only protein portion, but it is inactive by itself. That is known as the apoenzyme. Okay, we have a coenzyme or cofactor, which is a non-protein portion. The cofactor and coenzyme can also be there. So this coenzyme is the acts as the activator, and this complete apoenzyme or cofactor or coenzyme complex, which is known as the hollow enzyme. So this is a whole enzyme which is active, and it has the power to bind the substrate and to catalyze the particular transformation. So what are the definitions? So particularly definition of apoenzyme, it is the protein portion of the enzyme. Activity, it is inactive on its own and requires a cofactor to perform its catalytic function. Specificity, the apoenzyme is responsible for enzyme substrate specificity, ensuring it binds to the correct reactant. Heat sensitivity, as a protein, apoenzyme is sensitive to heat and can be denatured at high temperature. This is the typical characteristics of the proteins, right? An example, the protein component of DNA polymerase is an apoenzyme until it binds with the necessary metal ions to become active. Hollow enzyme, the definition, the complete complex formed by binding with of an apoenzyme and its required cofactors. Activity, it is fully functional and catalytically active form of the enzyme. Composition, it contains both protein part, apoenzyme, and the non-protein part, which is cofactor. For example, DNA polymerase, it is a complete form with all the necessary subunit cofactor. It is a hollow enzyme that replicates DNA. Carbonic anhydrase, the enzyme is a hollow enzyme when it contains the required zinc ion. And RNA polymerase, the complex that synthesizes RNA, is a well-known example of a hollow enzyme. Okay. So what are the difference between apoenzyme and hollow enzyme? Hollow enzyme refers to the apoenzyme along with the cofactor which is complete and catalytically active. Apoenzyme refers to an inactive form of the enzyme which activates upon binding to the cofactor. Inactive form of the enzyme is the apoenzyme. Catalytically active form is the hollow enzyme. Consists of the protein part of the enzyme. The hollow enzymes and consists of the protein part and and consist of the apoenzymes and one of the several cofactors. Incomplete enzymes that are less complex. Hollow enzymes are the complete and the complex enzyme. For example, catalytic components of DNA polymerase enzymes are considered as apoenzyme, and multi-subunit complex of DNA polymerase are known as a hollow enzyme. So there are some I think mistakes of the numbering of the points which you can recheck later. Okay. So this was the first question. Okay, so now the important thing is the cofactor component. A cofactor can be either a metal ion or a small organic molecule. Coenzyme description is a type of the organic cofactor that binds loosely to the apoenzyme. It acts as a temporary carrier of the functional groups or electrons during the reaction. Derived from many other coenzymes are derived from vitamins. For example, nicotinamide adenine dinucleotide or NAD plus and coenzyme A. Prosthetic group description, a cofactor that is tightly bound, sometimes covalently to the apoenzyme. It is not easily removed and is the integral part of enzyme's function. For example, the heme group of hemoglobin and the cytochrome protein, the flavin adenine dinucleotides, the derivative of vitamin B2, is a prosthetic group of the some enzymes. Okay, so this is the cofactor component, the cofactor which is essential for activating the apoenzyme and making of the complete hollow enzyme. Okay, so is there any doubt in the first question of today's session? Let me know if there is any doubt or if it is fine. If not, then we will move to the next problem. Anyways, you can let me know whether whatever doubt you have throughout our session in the chat box. I will discuss that at that time, or I will discuss it at the end of the session. Okay, it's fine. You can let me know about your doubts at any time of our session. Okay, through the chat box. Now let me go to the next problem of today's session. So problem number 2, it says that identify the incorrect statement among the following. Your options are: number A, acetyl-CoA is a thioester of coenzyme A and acetic acid. Number B, pyruvate is the starting point of the synthesis of acetyl-CoA. Number C, pyruvate is formed by metabolism of glycogen, and number D, TPP or thiamine pyrophosphate is the participates in the acetyl CoA formation. Which of the following statement is incorrect? Let me know. And read all the options very carefully. Okay, because whenever there is this kind of true false type of question, I find most of the students they make mistakes for this kind of question. So I want all of you to read the options very carefully, and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 2. Again, I will repeat the options. You have to identify the incorrect statement, right? Your option number A, acetyl-CoA is the thioester of coenzyme A and acetic acid. Number B, pyruvate is the starting point of synthesis of acetyl-CoA. Number C, pyruvate is formed by the metabolism of glycogen. And number D, TPP or thiamine pyrophosphate participates in the acetyl-CoA formation. Identify the statement that is incorrect. Read all the options carefully. Then let me know in the chat box A, B, C or D which of the following you think is the incorrect statement. See, even if your answer is wrong, it's fine, but please, whatever you think is the correct answer, let me know in the chat box. Otherwise, I won't understand if you are listening to me or not, or if you are in the class or not. Okay? Let me wait for a minute, then I will check the chat box. Identify the incorrect statement among the following. Number A, acetyl-CoA is a thioester of coenzyme A and acetic acid. Number B, pyruvate is the starting point of the synthesis of the acetyl-CoA. Number C, pyruvate is formed by the metabolism of glycogen. And number D, TPP or thiamine pyrophosphate participates in the acetyl-CoA formation. What will be the incorrect statement among these four? Let me check the chat box now. Okay, I got one answer which is number B. Let me check if number B is the correct answer or not. Okay, let me check. I have got only one answer which is number B. Let me check if it is correct or not. Okay, so the statement which is incorrect among the following will be, it is number C, which says that pyruvate is formed by the metabolism of glycogen. Okay, let me show you what is it all about. So first statement is says that acetyl CoA is a thioester of coenzyme A and acetic acid, which is correct. Why it is correct? So this is correct because we have this acetyl CoA. Okay, let me just erase this thing first. Okay, yeah. So if you can see, this is this blue portion. This comes from the acetic acid. Okay, and this all the other portion it comes from your coenzyme A. So this is a as you can see, this is a thioester because you know what is the structure of the ester. So for ester, we have this kind of structure. We have this carbon and carbon, then we have a O, and this comes from an alcohol. This comes from an acid. So as you can see, this comes from the acetic acid, and this all comes from your coenzyme A. Okay, so this is the thioester because this is formed by the, so normal esters are formed with the carboxylic acids and alcohol, and thioesters they form from the carboxylic acids and thiol. Okay, so the structure of the normal esters, so we know this is the we have this double bond O and we have a S here, and then this, as you can see, this is the thioester, the particular thing. Okay, so the statement number 1, which says that acetyl CoA is the is your thioester of coenzyme and acetic acid. This is correct. Then this comes the other questions. Okay, so basically acetyl CoA So in the case of acetyl-CoA metabolism, this is a acetyl-CoA is a central intermediate in the lipid metabolism. Okay, so we have this fatty acids which we have this acetyl-CoA, and then it goes to the ketone body. So you can see the precursors of the acetyl-CoA is the glucose. Glucose is converted to pyruvate followed by acetyl-CoA. Okay, so pyruvate basically is the starting point of the synthesis of the acetyl-CoA, and this pyruvate is formed by the metabolism of glucose, not the glycogen. Okay, and we have this amino acids are also involved in the in formation of this acetyl CoA. So which is, so this is important that you have to understand that acetyl CoA is the central intermediate of the lipid metabolism. Okay, and as you can see, it gives us the triglyceride, phospholipids, eicosanoids, carbon dioxide plus water plus energy, cholesterol, which give rise to the steroid hormones and bile salts. Okay, and one more thing we have to discuss it. Let me erase these things. So what is the end product of glycolysis? This is also important. Glyceryl 3-phosphate. It is converted to 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase, and the cofactors are involved here is the NAD plus PI, which gives the NADH plus H plus. 1,3 bisphosphoglyceraldehyde is converted to 3-phosphoglyceraldehyde, and this phosphoenol pyruvate is gives rise to the pyruvate by the pyruvate kinase. What is the fate of pyruvate? Pyruvate gives rise to the acetyl CoA by pyruvate dehydrogenase complex. We have the coenzyme A, which gives the carbon dioxide. NAD plus is involved. Okay, pyruvate plus ATP and carbon dioxide. This leads to the oxaloacetate, ADP, PI, and H plus. Pyruvate also gives rise to the acetaldehyde and ethanol. All and pyruvate converts to L-lactate by the enzyme lactate dehydrogenase. Okay, so these are the fate of the pyruvate. And thiamine pyrophosphate, yes, it is also participates in the acetyl CoA formation. So statement number C, it is false, which is said that pyruvate is formed by the metabolism as glycogen. No, it is formed by the metabolism of glucose. So is it fine, or do I need to repeat anything? Okay, if there is no doubt, I will go to the next problem. Okay. So the problem number 3, it says that what is the following coenzyme of the vitamin plantothanic acid? Your options are number A, pyridoxal phosphate, number B, tetrahydrofolate, number C, thiamine phosphate, and number D, coenzyme A. Which of the following is the coenzyme form of the vitamin plantothanic acid? Your options are number A, pyridoxal phosphate, number B, tetrahydrofolate, number C, thiamine phosphate, and number D, coenzyme A. Which of the following is the coenzyme form of the vitamin plantothanic acid? Again, I will wait for some time. I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer. Which of the following is the coenzyme form of vitamin plantothanic acid, pyridoxal phosphate, tetrahydrofolate, thiamine phosphate, or coenzyme A? I will wait for a minute, then I will check the chat box. Let me check the chat box. Let me check if I have any answer for the problem. Okay, I got two answers and both I'm saying it is number D. Now let me check if number D is the correct answer or not. So the correct answer is will be very good. It is number D, which is coenzyme A. Coenzyme A is the coenzyme form of the vitamin plantothanic acid. So this is the charts of the vitamin B complexes. So vitamin B1 or thiamine, it has a coenzyme thiamine pyrophosphate or TPP, and it is involved in the the group transfer, which is for which it is involved is a C2 aldehyde and decarboxylation. Vitamin B2 riboflavin, the coenzymes are flavin adenine mononucleotide or FAN and flavin diadenine dinucleotide or FAD. It is involved in the hydrogen transfer. We have vitamin B3 or niacin, which has nicotinamide adenine dinucleotide or NAD plus as a coenzyme. Nicotinamide adenine dinucleotide phosphate or NADP plus also now we have has a coenzyme and it is also involved in hydrogen transfer. And we have vitamin B5 or plantothanic acid, which has the coenzyme A and it is involved in the acyl group transfers. Okay, so it is very important. Plantothanic acid is coenzyme form is A and it is involved in the acyl group transfer. This is the vitamin B5. We have vitamin B6, which is your pyridoxine, and the pyridoxal phosphate is the coenzyme, and amino and decarboxylation is the group transfer. Vitamin B7, biotin, the coenzyme is the biocytin, and it is involved in the carboxyl group transfer. Vitamin B9, folic acid, the coenzyme is tetrahydrofolic acid, and it is involved in formal group transfer. Vitamin B12, cyanocobalamin, vitamin B12 coenzyme is the coenzyme, and carboxyl and HX rearrangements are the group and group transfer for which it is involved. So relationship between the plantothanic acid and coenzyme A. Plantothanic acid is a vitamin. There is a water soluble vitamin B. Soluble B vitamin. Plantothanic acid must be acquired from the diet by humans and other animals. Coenzyme CoA is a larger molecule. CoA is a complex biomolecule synthesized in a multi-step process from plantothanic acid, cysteine, and adenine triphosphate or ATP. CoA synthesis relies on the plantothanic acid. The initial and the rate limiting step of the CoA biosynthetic pathway is the phosphorylation of the plantothanic acid to the 4' phosphoplantothionate by the enzyme from plantothionate kinase. Okay, so this is important. Let me use the pen. Okay, so the CoA synthesis relies on the initial rate step of CoA biosynthesis pathway is the phosphorylation of the plantothanic acid. The phosphorylation of the plantothanic acid to the 4' phosphoplantothanate by the enzyme plantothionate kinase. CoA structure, which is incorporates the plantothanic acid as a final CoA molecule, contains a plantothanic acid moiety along with the adenosine diphosphate and the cysteamine. The function of the CoA depends on the synthesis of the plantothanic acid. CoA is the major acyl group carrier when its most important derivatives being acyl CoA and fatty acyl CoA. Without a sufficient supply of plantothanic acid, an organism cannot synthesize the CoA, which disrupts the vital metabolic pathways. So this is the synthesize how the CoA enzyme A is synthesized from plantothanic acid. So this is a 5-step process. As you can see, first step is the phosphorylation step, where the plantothanic acid is converted to the 4' phosphoplantothanate acid by the enzyme plantothanic acid kinase, and here ATP is converted to ADP. Then it is inhibited by the CoA and CoA derivatives. Okay, inhibition is reversed by the cartimine. Okay, so this this is the first and most regulated step. Second step is the cysteine addition step. A cysteine molecule is added to the 4' phosphoplantothanate acid and converts it to the 4' phosphoplantothanate pantothenyl cysteine by the enzyme 4' phosphopantothenol cysteine synthase. Then third step is the decarboxylation step, where cysteine portion is decarboxylated to form the 4' phosphoplantothane by the enzyme 4' phosphoplantothane cysteine decarboxylase. Then the fourth step is the adenylation. Adenosine monophosphate or AMP is added to the adenosine to the 4' phosphoplantothane to form the 4' D-phospho coenzyme A, and the last step is the final step is the phosphorylation of the 4' D-phospho coenzyme A to the coenzyme A by D-phospho CoA kinase. ATP is converted to ADP here. As you can see, coenzyme A also can be converted to 4' phospho cycloplantotheniene by the CoA hydrolase. Okay, so these are the summary of the synthesis pathway of the plantothenic acid to coenzyme A. What is the biological significance? This is important, which you have to remember. The biological significance, the relationship is critical for the cellular energy production, and acyl CoA as acyl CoA, CoA is a key entry molecule for the citric acid cycle, allowing the body to break down the carbohydrate, fats, and proteins for energy. Biosynthesis of essential molecule, it is required for the synthesis of lipids, steroid hormones, cholesterol, carrier protein, which is necessary for fatty acid synthesis. Protein modification, the transfer of acyl groups by CoA is an important metabolism for the regulating function of the many proteins. Okay. So yeah, your answer was correct, which is given by you. So is it clear, or do I need to repeat anything? The biological pathway, if you can remember this conversion, which is very good because it is very important. You do not need to remember the structure, but this biological, I mean this pathway, this 5-step reaction, it will be better if you can remember it. Okay. So if there is no doubt, I will move to the next question. Anyways, you can let me know later also, whatever doubt you have, we can discuss it at the end of the session. That is also possible, or you can let me know if you have any problem. Okay. I will then move to the next problem. So problem number 4, it says that which of the following statements is true regarding the folic acid biosynthesis? Your options are: number A, it consists of a pyridine ring, PABA, and aspartic acid. Number B, the conversion of dehydroteroic acid to dehydrofolic acid is converted by dehydrofolate synthesis enzyme. Number C, the conversion of dehydroteroic acid to dehydrofolic acid is converted to dehydrofolate reductase enzyme. Number D, humans can biosynthesize folic acid. Which of the following statements is true regarding the folic acid biosynthesis? Again, I will repeat. Number A, it consists of a pyridine ring, PABA, and aspartic acid. Number B, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthetase enzyme. Number C, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate reductase enzyme. Number D, humans can biosynthesize folic acid. Which of the following statement is true regarding the folic acid biosynthesis? Again, I am telling, since it is a true false type of question, please be careful, read options very carefully. Then let me know in the chat box A, B, C, or D which of the following you think is the correct answer for problem number 4. I will wait for a minute, then I will check the chat box. Which of the following statements is true regarding the folic acid biosynthesis? Which of the following statements is true regarding the folic acid biosynthesis? Let me check the chat box now. Let me check if I have any answer. I got two answers. One is number C and another is number B. Let me check which of the following is the correct answer. So the correct answer, the statement which is true regarding folic acid biosynthesis will be It is number B. The conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by the dihydrofolate synthesis enzyme. So this is the structure of folic acid. Okay, the folic acid is actually a component. It consists of the pteridine ring. This is the structure of a pteridine ring. We have a para-aminobenzoic acid or PABA. Then we have a glutamic acid. The pteridine ring and para-aminobenzoic acid together, it is called the pteroic acid, and together this is called the pterodyl glutamic acid or folic acid. So this is the conversion. So para-aminobenzoic acid and pteridine ring, and so this converts to the dihydrofolic acid by dihydrofolate synthesis enzyme. Then dihydroteroic acid converts to dihydrofolic acid by dihydrosynthesis, dihydrofolate synthesis, and this dihydrofolate is converted to tetrahydrofolate, which is the active form of folic acid by the dihydrofolate reductase. Okay. So the statement number C, number B, which says the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis. It is the correct answer. Okay. So is it clear, or do you have any doubt? Then I will repeat this. Okay, I will repeat it. Okay. So just let me show you one more time. So basically this folic acid metabolism, it is a very crucial thing which happens in our body. I will also discuss it in detail in our upcoming slides. Then you will also get a better picture of this particular point. So the statements, look at the statements very carefully. So the statement number A, it says it consists of a pteridine ring, PABA and aspartic acid, which is correct, which is false because you can see the structure. We have this pteridine ring, PABA and the glutamic acid. Okay, so aspartic acid is not there. There is a glutamic acid. So that is the statement is false. Number B, it says the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthase enzyme. So whatever I have showed you. So this is the metabolism. What are the structures and all these things? These I will just show you later because I will discuss it in detail. Okay, so para-aminobenzoic acid or pteridine ring, this is give rise to the dihydroteroic acid. Okay, so this dihydroteroic acid, the enzyme which is catalyzes this step is the dihydroteroid synthesis enzyme. Then dihydroteroic acid is converted to dihydrofolic acid, and the enzyme which catalyzes is the dihydrofolate synthesis. It is very easy to remember. So suppose you do not remember exact what, or you are confused what is the name of the particular correct enzyme. So you can say dihydroteroic to dihydrofolic acid is catalyzed by dihydrofolate synthase. Synthase means the enzymes which will synthesize a particular component. Dihydrofolate synthase means it will synthesize the dihydrofolic acid, whereas the conversion of the statement number C, which says it is converted by dihydrofolic reductase, it is incorrect. Reductase means the enzyme which will reduce. So dihydrofolate converted to tetrahydrofolic. So it also seems like this is a reduction kind of step, where dihydrofolate reductase is the enzyme which reduces dihydrofolic to tetrahydrofolic. Okay, so that is why statement number C is false, and number D, which says humans can synthesize folic acid, it is also false because humans cannot biosynthesize the folic acid. So that is why statement number B is true and it is the correct answer. Now is it clear, or do I need to tell you more? Okay, okay. Thank you. For if you have any doubt, you can keep asking. Okay. So I think it is clear. I will show you what is the particular structures I will discuss in detail in our upcoming slides. So I think you will get a better picture in our upcoming slides regarding this biosynthesis of folic acid. Okay. So now I will go to the next problem. Problem number 5, it says pyruvate carboxylase uses dash as a prosthetic group. Your options are number A acetyl-CoA, number B retinol, number C ascorbic acid, number D biotin. Pyruvate carboxylase uses dash as the prosthetic group. What will be the correct answer? Acetyl-CoA, retinol, ascorbic acid, or biotin? Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which is the correct prosthetic group for pyruvate carboxylase. Pyruvate carboxylase uses dash as the prosthetic group. What will be the correct answer? Let me check the chat box. I think I will get some answers there. Okay, I got one answer which is saying it is number D. Okay, let me check if number D is the correct answer or is there any other answer. So, pyruvate carboxylase, what does it use as the co-prosthetic group? The correct answer is yes, it is number D. Biotin is the prosthetic group for the pyruvate carboxylase. So, this is the structure of biotin. Okay. So biotin acts as a for pyruvate carboxylate is biotin is acts as a prosthetic group for this particular enzyme. So what does this pyruvate carboxylase do? Pyruvate carboxylase basically converts pyruvate to oxaloacetate. As you can see, this is the conversion. This is the step. So it is adding up a carbon dioxide group to to this pyruvate to make it oxaloacetate, and bicarbonate and ATP is converted to for ADP and PI. Okay, so how does it work? So as you can see, this is the structure, and then the pyruvate it comes here. At first, this particular group it abstracts the proton abstracts the hydrogen from this methyl group, and there is this kind of intermediate. This kind of intermediate is formed. There we have carboxylic acid here, which is added to our to form the pyruvate to oxaloacetate. So this is the reactions. Okay, this is a very easy one or two step reaction. So I hope you will practice and you can do it easily by yourself. One or two time you have to, I think, practice. So pyruvate carboxylase uses biotin as a prosthetic group. So this thing is important that biotin has a 5-carbon side chain whose terminal carboxyl is an amide linkage to the epsilon amino group of the enzyme of an enzyme lysine. Okay, so this is important which you have to remember. So biotin has a 5-carbon chain, this one, which terminal carboxylase is in linkage with the epsilon amino group of the enzyme lysine. The biotin and lysine side chains form a long swinging arm that allows the biotin ring to swing back and forth between two active sites. Okay, so as you can see, this is the lysine residue, this is the terminal carboxylic group of the biotin, and this is the epsilon amino group of the lysine, and it forms a swinging arm. This allows biotin ring to swing back and forth. Okay, this is the structure of lysine. If you have forgotten, this is the structure of lysine which is given. Okay, so I hope it is fine. Or do you have any doubt in this question? Okay, let me know if you have any doubt. I will repeat it one more time. If not, let me proceed to our next question. So problem number 6, it says that which co-substrate is involved in the conversion of hydroxyproline from proline? Your options are number A 2-oxoclitoglutarate, number B coenzyme A, number C NAD+, and number C GABA. Which co-substrate is involved in the conversion of hydroxyproline from proline? Number A 2-oxaloglutarate, number B coenzyme A, number C NAD+, and number D GABA. I will give you a minute or two, then I will check the chat box. Let me know which co-substrate is involved in the conversion of hydroxyproline from proline. Which co-substrate is involved in conversion of hydroxyproline from proline? Number A 2-oxoglutarate, number B coenzyme A, number C NAD+, number D GABA. I will wait for 1 or 2 minutes, then I will check the chat box. Which co-substrate is involved in the conversion of hydroxyproline to proline from proline? Whatever you think, let me know in the chat box, A, B, C or D, whatever you think is the correct answer. As I have said, if even if your answer is wrong, it is fine. Which co-substrate is involved in conversion of hydroxyproline from proline? Let me now check the chat box. Okay, I got true answers and both are saying it is number A. Let me check if number A is the correct answer or something else. So the co-substrate that is involved in the conversion of hydroxyproline from proline will be, yes, very good. It is number A, 2-oxoglutarate. So this is the structure of proline. Proline is converted to hydroxyproline. There is only the addition of a hydroxyl group to this carbon of the proline. As you can see, proline hydroxylase is the enzyme that is involved. So, okay, so along with the name of the co-substrate, you have to remember what is the name of the enzyme. It is the proline hydroxylase, right? And what is the co-substrate? Co-substrate 2-oxoglutarate, which is converted to succinate. Okay, and also there are some side reactions happens like oxygen and Fe2 plus is converted to Fe3 plus and carbon dioxide. This Fe is present in this enzyme and vice versa, and also vitamin C is converted to the dehydroascorbate in this step. Okay, in this particular step. So what is the process of the proline hydroxylation? The conversion of the proline to hydroxyl proline is catalyzed by the enzyme prolyl 4-hydroxylase or P4H, or we can the proline hydroxylase is also is correct. The reaction occurs inside the endoplasmic reticulum and involves these steps. First is the protein synthesis. The amino acid proline is incorporated into newly synthesized polypeptide chain such as procollagen by ribosomes. Then number 2 is recognition. The P4H enzyme specifically recognizes and binds to the protein residues located in a repeating X-pro-gly sequence within the polypeptide chain. Number 3, hydroxylation reaction. Using its catalytic site, the P4H enzyme introduces a hydroxyl OH group into the fourth carbon of the proline ring, creating hydroxyproline. Number 4, cofactor requirements. The reaction is dependent on 4 key cofactors and substrates like molecular oxygen, ferrous ion, alpha-ketoglutarate and ascorbate. Molecular oxygen provides the oxygen atom for the hydroxyl group. So remember this hydroxyl group comes from the molecular oxygen. Okay, ferrous ion is essential meta-ion for the enzyme's active site. Alpha-ketoglutarate is a co-substrate that is converted to the succinate and carbon dioxide during the reaction. And number 4, ascorbate or vitamin C acts as a reducing agent to keep the iron in its functional or ferrous to a state. Without vitamin C, the enzyme cannot function properly and the synthesis of the stable collagen is impaired. So these things you have to remember. What are the important key cofactors and the important key cofactors? Molecular oxygen, we have the ferrous ion, which is important. Molecular oxygen is important. 2-oxoglutarate or alpha-ketoglutarate is also important, and we have vitamin C or ascorbic acid. These play a very crucial role in the for the conversion of the proline to the hydroxyproline. And what is the significance of hydroxyproline? So why hydroxyproline is so much important? Because the conversion of a proline to hydroxyproline is essential to function and stability of collagen. Okay, triple helix stability in addition to the hydroxyl group of the proline allows the formation of a stable 3-stranded collagen triple helix by the defining structure of the collagen. Structural strength, the stability of the triple helix gives the connective tissues like skin, cartilage and bone their tensile strength and rigidity. Disease prevention, the lack of vitamin C leads to the defect in the proline hydroxylation resulting in unstable collagen. This causes scurvy, a disease characterized by weakened connective tissue, hemorrhoids and impaired wound healing. Okay. So these are the significance of the your hydroxy proline in the collagen. So is it clear, or do I need to repeat anything in this question? Let me know if it is clear, or if I need to repeat anything. Yes, A was the correct answer. Okay, so I think it is clear. Now let me go to the next problem. So problem number 7, it says that sulfonamides are dash inhibitor for the dihydropteroid synthesis. Your options are number A reversible, number B irreversible, number C suicide, and number D ASMII. Sulfonamides are which kind of inhibitor for dihydropteroid synthesis? Again, I will wait for 1 or 2 minutes, then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer. Sulfonamides are dash inhibitor of dihydropteroid synthesis, reversible, irreversible, suicide, or ASMII. Sulfonamides are dash inhibitor of dihydropteroid synthesis. What is the correct answer? Reversible, irreversible, suicide, or ASMII? I will wait for another minute, then I will check the chat box. Whatever you think, let me know. A, B, C, or D, which of the following is the correct answer? Let me check the chat box. Let me check if there is any answer for problem number 7. Okay. I got only one answer which says it is number D. Let me check if number D is the correct answer or not. So the correct answer will be, it is number A. Sulfonamides are the reversible inhibitor of the dihydropteroid synthesis. Okay. So this is the folate metabolism. The conversions I have discussed earlier also. Pteridine or dihydropteroid diphosphate plus para-aminobenzoic acid converts to the dihydropteroic acid or DHP or dihydropteroid. Then dihydropteroid is converted to dihydrofolate by the dihydrofolate synthesis enzyme, and this dihydrofolic acid or dihydrofolate converted to tetrahydrofolic acid or tetrahydrofolate or THF by the enzyme dihydrofolate reductase. Sulfonamides, they act as a reversible inhibitor of the dihydropteroid synthesis, and dimethoprim, they acts as the inhibitor for the dihydrofolate reductase. So what is the particular, these are the steps, these are the structures I have already told you that I will show you later, or whoever has a doubt in this particular question I have mentioned. So as you can see, this is the pteridine, okay, or the dihydropteroid diphosphate, and para-aminobenzoic acid converts the dihydropteroid. Okay, so this is the structures. There I haven't given any structure, here I have mentioned the structures. So this is the structure. As you can see, this is nothing but this is the pteridine which you have to remember, and this is the para-aminobenzoic acid. Only there is a this kind of I mean elimination of this hydroxyl group, and it forms a dihydropteroid or dihydroteroic acid, you can also say. So here you can see sulfonamides are present here, which acts as a reversible inhibition. So why sulfonamides, they act as a reversible inhibitor? Reversible inhibitor, so because before knowing that, we have to identify what is reversible and what is the irreversible inhibitor. So reversible inhibitors, the enzyme do follow Michaelis-Menten rate equation and Lineweaver Burke plot. Okay, the plot V and S curve are rectangular hyperbolic shape, dissociate very rapidly from the targeted enzyme because they're loosely bound. Classified into three categories: competitive, non-competitive, and mixed inhibitor. Competitive inhibitors can be reversed by increasing substrate concentration. Reversible inhibitors bind to the enzyme by hydrogen bonding and hydrophobic interaction and the ionic bonds. So basically, the sulfonamides, their structures are very much similar to their the para-aminobenzoic acid. Okay, that is why it acts as a reversible inhibitor for this particular enzyme dihydrofolate synthesis. But increasing the amount of the para-aminobenzoic acid can reverse the action of this sulfonamides, means the binding of the sulfonamide to this dihydroteroic synthesis enzyme is reversible. That is why it is a reversible inhibitor. And what are the irreversible inhibitors? They do not follow Michaelis-Menten equation. The plot V and S are sigmoidal curve, dissociate very slowly from the targeted enzyme because they are tightly bound. Classified into three categories: root specific reagents, substrate analogs, and suicide inhibitors. Substrate analogs imitate enzyme substrate and irreversibly modify the active site of the enzyme. Irreversible inhibitor binds to the enzyme through the covalent interaction, which modify amino acid residues by the reactive functional group. Dihydroteroid is converted to dihydrofolate by the L-glutamic acid. It is the structure of the you can see this is the structure of the dihydrofolate. Okay, and then this particular your dihydrofolate is converted to tetrahydrofolate or the coenzyme F or the folic acid, you can say. Okay, by this particular enzyme, and here trimethoprim acts as a inhibitor of this dihydrofolate reductase. So this is the mechanism of actions of the sulfonamides. So is it clear? It might seem like a little difficult with the structures. They may seem little bit hard, but if you practice it one or two time, I think it will be clear to you, because it is not very much hard. It is quite easy, but you have to practice it by writing on your copy. Otherwise, you won't remember these things properly. Okay. So if there is no doubt, I will move to the next problem. So problem number 8, it says that the roles of TPP and lipoic acid in formation of acetyl-CoA. Your options: number A, decarboxylation and transesterification; number B, transesterification and oxidation. So this is the respectively. Okay. The first ones are the roles of TPP, and the second options are for the roles of the lipoic acid. So what are the roles of the TPP and lipoic acid in formation of acetyl-CoA respectively? Number A, decarboxylation and transesterification; number B, transesterification and oxidation; number C, dehydrogenation and alkylation; number D, decarboxylation and transamination. What are the roles of TPP and lipoic acid in the formation of acetyl-CoA? What are the roles of TPP and lipoic acid in formation of acetyl-CoA? What are the roles of TPP and lipoic acid in formation of acetyl-CoA? Number A, decarboxylation, transesterification; number B, transesterification and oxidation; number C, dehydrogenation and alkylation; number D, decarboxylation and transamination. What are the roles of TPP and lipoic acid in formation of acetyl-CoA? I will wait for a minute, then I will check the chat box. The roles of TPP and lipoic acid in formation of acetyl-CoA. Let me check the chat box. Okay, I got two answer. Both are saying it is the number A, which is the correct answer for problem number 8. Let me check if number A is the correct answer or not. So the correct answer will be yes, it is number A. Decarboxylation and transesterification are the particular roles of the TPP. The role of TPP is the decarboxylation, role of lipoic acid is the transesterification. So basically, this is the mechanism of the pyruvate dehydrogenase enzymes reaction, and this is for the formation of the your acetyl CoA. So I have already mentioned that acetyl CoA converts to the pyruvate is converted to the acetyl CoA in our very second question, I guess, in today's session. So this is the complete mechanism how it happens. So this step number one this is the structure of pyruvate. So pyruvate first loses the carbon dioxide, and it forms the hydroxyethyl TPP, the hydroxyethyl TPP or hydroxyethyl or HETPP. Okay, and this HETPP is converted to our thiamine for pyrophosphate. Okay, so step one is the decarboxylation of the pyruvate, which occurs with the formation of the hydroxyl TPP. Okay, this is the step one. And then the step this is the thiamine pyrophosphate, which facilitates this step one. The TPP means thiamine pyrophosphate, right? So thiamine pyrophosphate converted to hydroxy TPP, or then the step two is the step two is the transfer of the 2-carbon unit to the lipoic acid. Okay, so as you can see, this particular group, which is in the blue here, which is comes from the pyruvate, it is added to the this is the lipoic acid. You can see, so it is added to this thiol group, so it forms a thioester with the lipoic acid. So this alcohol part, it goes here, and it forms a thioester with the lipoic acid, and then this our coenzyme A, it comes and it forms the acetyl CoA. Okay, so the first step is the pyruvate loses carbon dioxide and HETPP is formed. Step 2, hydroxyethyl group is transferred to the lipoic acid and oxidized to form acetyl lipoamide, and then acetyl group is transferred to the CoA to form the acetyl CoA, and then hydroxypyruvate, it is deoxidized. So this thing will be deoxidized, and these are the enzymes which are involved. Pyruvate dehydrogenase catalyzes the first step. The second step is catalyzed by the dehydrolipolyl transacylase, and which catalyzes this transesterification process. And then last step is the dehydrolipoid is reoxidized by the dehydrofolate dehydrogenase. Okay, so the lipoic acid is deoxidized, and this is the step 4 of this particular reaction. So what is the role of TPP and lipoic acid? So TPP and lipoic acid are the crucial coenzymes in the pyruvate dehydrogenase complex, which catalyzes the conversion of the pyruvate to acetyl CoA, linking the glycolysis to the citric acid cycle. The role of the TPP is the coenzyme of pyruvate dehydrogenase E1 subunit of the PDC, or the pyruvate dehydrogenase complex. It catalyzes the oxidative decarboxylation of pyruvate, meaning it removes the carboxyl group from pyruvate as a carbon dioxide and simultaneously oxidizes the remaining 2-carbon unit. TPP forms a covalent intermediate hydroxyethyl TPP with the 2-carbon fragment derived from pyruvate, thus initiating the reaction. Role of the lipoic acid. Lipoic acid, specifically in its deoxidized disulfide from lipomide, is covalently attached to the lysine residue of the dehydrolipolyl acylase or E2 subunit of the PDC. It functions as a swinging arm that carries the acyl group from E1 of active side of the E2. The lipoic acid undergoes reductive acylation, accepting the acyl group from hydroxyethyl TPP or HETPP, becoming S-acetyl dihydramide while becoming reducing the dithiol form. Subsequently, the acyl group from S-acetyl dihydrolipomide is transferred to the coenzyme A by E2, forming acetyl CoA. The reduced dihydrolipomide is then reoxidized by dehydrolipolyl dehydrogenase or E3 subunit, regenerating the oxidized lipomide for further catalytic cycles, with the NAD plus being reduced by NADH in the process. Okay, so these are the roles of TPP and lipoic acid. So is it clear, or do I need to repeat the roles of the TPP and lipoic acid again? Okay, I think there is no doubt in this particular question. Then I will move to the next problem. Okay, now let me go to the next problem of today's session. If there is no doubt in this question. Okay, now let me go to the problem number 9. So problem number 9, it says, which of the following statements is false? Your options: number A, sulfadiazine is a bacteriostatic in nature. Number B, sulfadiazine is bactericidal in nature. Number C, sulfadiazine may show side effects like nausea, dizziness, blood disorders, etc. And number D, sulfadiazine may be administered orally or topically. Which of the following statement is false? Number A, sulfodiazine is bacteriostatic in nature. Number B, sulfodiazine is bactericidal in nature. Number C, sulfodiazine may show side effects like nausea, dizziness, blood disorder, etc. And number D, sulfodiazine may be administered orally and topically. Let me know A, B, C or D, which of the statement you think is false regarding the sulfodiazine. Again, I will repeat problem number 9. Which of the following statements is false? Number A, sulfadiazine is bacteriostatic in nature. Number B, sulfadiazine is bactericidal in nature. Number C, sulfadiazine may show side effects like nausea, dizziness, and blood disorders, etc. Number D, sulfadiazine may be administered orally or topically. Let me wait for a minute, then I will check the chat box for your answers. Which of the following statement will be false? Let me check the chat box now. Okay. So I got two answers. Both are saying number B is the correct answer. Let me check if number B is the correct answer or not. So the statement which is false will be very good. It is number B. Sulfadiazine is bactericidal in nature. Okay. So what is a bacteriostatic? What is a bactericidal? I will discuss it in detail in our upcoming sessions when we will discuss about the antibacterial drugs. But just let me tell you that bactericidal is the components of which entirely kills the bacteria, and bacteriostatic is the components or the antibacterial agents which actually inhibits its growth but do not kill it entirely like the bactericidal. Okay. So number B is the false statement. So sulfadiazine is a sulfonamide antibiotic used to prevent and treat various bacterial infection, and is also contains some parasitic infection. It is available as an oral tablet, also in a topical form like silver sulfadiazine for burn wound infection. Uses sulfadiazine treats the range of the bacterial infections using urinary tract infections, ear infections, lung infection like pneumonia, bronchitis, intestinal infection, bacterial meningitis, sexually transmitted infections like the chancroid and chlamydia. Is also used for the parasitic infections such as the toxoplasmosis, also combination with the pyrimethamine. Contains types of certain types of malaria as the adjuvant therapy of the chloroquine resistant strains. Topical silver sulfadiazine is specifically used for preventing and treating wound infections in second or third degree burns. Mechanism of action: sulfadiazine works by inhibiting growth of the bacteria, which is means it is the bacteriostatic. It achieves this by interfering with the folic acid metabolism. Bacteria require folic acid for the synthesis of nucleotides, and which are essential for DNA and RNA production. Competitive inhibition of the dihydropteroid synthesis. Sulfadiazine structure mimics the para-aminobenzoic acid or PABA, is a substrate for the bacterial enzyme dihydropteroid synthesis. By competing with the PABA, it blocks the enzyme's activity, thus preventing the function of the dihydrofolic acid, a precursor of tetrahydrofolate, preventing the tetrahydrofolate formation. This disruption of the dihydrofolate synthesis ultimately impedes the formation of the tetrahydrofolate, which is necessary for bacterial DNA and RNA and protein synthesis, leading to the inhibition of the bacterial growth and proliferation. Side effects. Common side effects are the oral sulfadiazine can include nausea, vomiting, diarrhea, stomach pain, loss of the appetite, headache and dizziness and trouble sleeping, skin reactions like rash, increased sensitivity to sunlight. Serious but less common side effects that require immediate medical attention include several allergic reactions, blood disorders, okay, like indicated by fever, sore throat, unusual bleeding, bruising, pale skin, unusual weakness, kidney problems indicated by the severe pain in the side or back, and blood in urine, liver problems like jaundice, upper stomach pain, and dark urine. So these are the side effects. Okay. So is it clear or is there any doubt regarding the sulfadiazine? Okay, I hope it is clear. And yes, your answers were correct. So very good. Now let me go to the next problem. So problem number 10, it says which of the following enzyme is involved in the conversion of DUMP to DTMP? Your options are number A SHT, number B DHFR, number C DHFS, and number D TS. Which of the one of the following enzyme converts DUMP to DTMP? See, most of the cases, the questions may are given by using the complete names of the enzyme, but sometimes the abbreviations can also be used. Okay, sometimes the question can give you only the abbreviations, but you have to just think and you have to identify what is the full form of that particular abbreviation and then answer the particular question. Okay, not that all of the time you will get the complete terms. Sometimes you will get the abbreviations. So reading or remembering the abbreviations are equally important. Which one of the following enzyme converts DUMP to DTMP? I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer. Even if your answer is wrong, it is fine. So which of the following enzyme in converts DUMP to DTMP? Let me check the chat box. Let me check. Okay, I got one answer which is saying it is number B. Okay, let me check if number B is the correct answer or not for problem number 10. So the correct answer will be, it is number D. Okay, number D is the correct answer. So TS means your thymidylate synthase. Okay, so this enzyme converts the DUMP to DTMP. So this is the DUMP and this is the DTMP, right? So U is indicates the uracil. As you can see, uracil, it is a uracil is present here, and T is the thymine. So the difference of thymine and uracil is thymine has a methyl group in this position. Otherwise, the structure is same, right? The thymidylate synthesis, what does it do? It converts the DUMP to DTMP, and this methyl, from where this methyl group generates, we have a side reaction which happens, like if we have N5 N10 methylene tetrahydrofolate, and which converts to 7A dihydrofolate, and here you can see there is a hydrogen in this place and there is a carbon in this place, which CH2, and these are forms the methyl group in the this DTMP. So thymidylate synthase, or TS, is a crucial enzyme in cellular biology, primarily known for its essential role in DNA synthesis and repair. It has gained a significant attention in the medical research, particularly in the field of the cancer chemotherapy, due to its central position in the production of DNA building blocks. The function and mechanism of action catalyzing the DTMP synthase, thymidylate synthase catalyzes the reductive methylation of the deoxyuridine monophosphate, or DUMP, to deoxythymidine monophosphate, or DTMP, essential for the DNA synthesis. DTMP is then further phosphorylated to deoxythymidine triphosphate, or TTP, which is crucial precursor for the DNA replication and repair. Therefore, TS provides the sole de novo pathway for the production of the thymidylate, a critical component of the DNA. Cofactor involvement: the reaction requires specific cofactors. The 5,10 methylene tetrahydrofolate, which acts as a both one carbon donor, providing the methyl group subsequently as a reductant during the catalytic process. Importance in the DNA synthesis and repair: maintaining DNTP balance. TS plays a vital role in regulating the balanced supply of the four DNA precursor DTNPs. Proper nucleotide pool balance is essential for accurate DNA replication and minimizing the DNA damage. Preventing timeless death. Inhibition of TS leads to the depletion of DTMP and consequently DTTP. This imbalance can severely disrupt the DNA synthesis and repair, leading to the phenomenon known as the timeless death, which is formed of the programmed cell death or apoptosis during the in the actively divided cell. Okay. So these are all about the thymidylate synthesis, or TS enzyme, which is a very crucial component of the cellular biology. Okay. So is it clear what is the role of the TS and how the conversion happens? So the side reactions are equally important. As you can see, the conversion of DUMP to DTMP is important, and the other conversion, the N5 N10 methylene tetrahydrofolate to 7A dihydrofolate, is also important, because this conversion provides the methyl group in the DUMP to DTMP. Okay. So I hope it is clear. If not, you can let me know in the chat box. Otherwise, I will go to the next problem. So problem number 11, it says, which of the following is an example of the TS inhibitor? TS is a thymidylate synthesis, which we have discussed in our last question. So which of the following is an example of a TS inhibitor? Your options are: number A, camptothecin; number B, cisplatin; number C, raltetrexate; and number D, atorvastatin. Which of the following is an example of the TS inhibitor? Camptothecin, cisplatin, raltetraxate, and atorvastatin. I will wait for a minute or two, then I will discuss this. Let me know A, B, C, or D, which of the following is the correct answer for problem number 11. Which of the following is a TS inhibitor? Camptothecin, cisplatin, raltetrexate, atorvastatin. Which of the following is an example of TS inhibitor? Let me know. Which of the following is an example of TS inhibitor? Let me check the chat box. I have two answers. Both are saying it is number D. Let me check if number D is the correct answer or not. So, the compound or the drug which is an example of a TS inhibitor will be, it is actually number C, raltetrexate. It is an example of TS inhibitor. Okay. So camptothecin is an anti-cancer drug and it is a topoisomerase inhibitor. Cisplatin, it works by other mechanisms. Okay. It is not a particular enzyme inhibitor. Raltetrexate is a TS inhibitor. Atorvastatin is actually, these are the statin class of compound which are the HMG-CoA inhibitors which acts as an anti-cholesterol drug or cholesterol lowering drug. We will discuss we will discuss about this camptothecin, cisplatin, atorvastatins. All we will discuss in our upcoming sessions because it is in our course. It is in your course, but not in today's class. Okay. So raltetrexate is an example of TS inhibitor. So the TS inhibitors is a drug. TS inhibitor is a drug that blocks the action of the thymidylate synthesis enzyme, preventing the synthesis of deoxythymidine monophosphate or DTMP, a crucial component of DNA. By disrupting DNA synthesis, these inhibitors halt the cell division and are used as an effective strategy in cancer chemotherapy, particularly against fast-growing tumor cells, and are also sometimes used in combination with other cancer treatment. How it works? It targets the TS or thymidylate synthase inhibitor, binds to and blocks the activity of thymidylate synthase enzyme, blocks the DTMP synthesis. This blocks the methylation of the deoxyuridine monophosphate or DUMP to DTMP, which is essential for the creation of thymidine, impairs the DNA synthesis. Without enough DTMP, cell cannot produce necessary nucleotides to build new DNA. Induces cell death. It leads to the halt of the DNA synthesis, DNA damage, and ultimately the apoptosis or programmed cell death in the tumor cell. Examples of the TS inhibitors: 5-fluorouracil or 5-FU, a widely used fluoropyrimidine that is converted to active metabolite FDUMP, which then inhibits TS. Raltetrexate, a folate analog approved for the advanced colorectal cancer in several countries. Pemetrexate, another antifolate agent that has shown promise in various solid tumors, including the mesothelioma. And capecitabine, an oral furosemide design. Fluoropyrimide is designed to convert it to 5-FU, preferentially within the tumor cell. And this is the structure of your raltetrexate. You do not need to remember the structures of these all. Just remember the names of the drugs. Okay, like this 5-fluorouracil, raltetrexate, pemetrexate, and cepetabine. Okay, so just remember the names. You do not need to memorize the structure. I just have shown you for your understanding. Okay. Now, let me go to the next problem. Problem number 12, which says, which of the following statements is false regarding ascorbic acid? Your options are: number A, it is a strong antioxidant. Number B, it catalyzes hydroxylation reaction. Number C, it is produced inside human body, not required through diet. Number D, it chelates with the iron to make it more soluble. Which of the following statements is false regarding ascorbic acid? Again, I am repeating since it is a true-false kind of question. Read all the options very carefully and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 12, or which statement is false regarding the ascorbic acid. Number A, it is a strong antioxidant. Number B, it catalyzes hydroxylation reaction. Number C, it is produced inside human body, not required through diet. And number D, it chelates with the iron to make it more soluble. I will wait for a minute or two, then I will check the chat box. Meanwhile, let me know A, B, C or D which of the following you think is the correct answer for problem number 12. Which of the following statements is false regarding ascorbic acid? Which of the following statements is false regarding the ascorbic acid? Number A, it is a strong antioxidant. Number B, it catalyzes hydroxylation reaction. Number C, it is produced inside the human body, not required through diet. Number D, it chelates with the iron to make it more soluble. Let me check the chat box. The slides are visible now, right? Let me know. Okay. So there might be some kind of internet network issue. So I got one answer, which is number C. Let me check if number C is the correct answer or not for our problem number 12. Let me see. So the statement which is false regarding the ascorbic acid will be, yes, it is number C, which says it is produced inside our body, not required through diet. No, ascorbic acid is not produced in our body. It is required to acquire it by the diet. Okay, so ascorbic acid, commonly known as vitamin C, this is the structure, is an essential water-soluble vitamin that humans must obtain through their diet. It is a potent antioxidant crucial for many physiological processes, including the production of collagen, a protein essential for protein, skin, bones, and connective tissues. So biological functions, collagen synthesis. Ascorbic acid is a cofactor for the enzymes involved in the hydroxylation of proline and lysine residues, which is a key step in forming stable triple helix structure of collagen. If you remember, when I was discussing the conversion of proline to hydroxyl proline, I have mentioned that ascorbate is a side cofactor, and which is very crucial because it maintains the iron plus 2 state, which is extremely crucial for the enzymes activity. Okay, so this is, and as an antioxidant, it protects the body from oxidative stress by scavenging reactive oxygen species and regenerating other antioxidants such as vitamin E. Iron absorption, vitamin C enhances the absorption of non-heme iron, the type of iron found in plant foods. Immune function. The vitamin plays an important role in immune function with high concentrations found in immune cells. Neurotransmitter synthesis. It is a coenzyme for dopamine beta hydroxylase, which converts the neurotransmitter dopamine to norepinephrine. The hormone synthesis. It is needed for the synthesis of the cortisol in the adrenal glands. Wound healing by supporting collagen formation. Ascorbic acid is vital for repair, for the repair of the tissues and the healing of the wounds. Okay. So these are the crucial biological functions for the ascorbic acid. So is it clear, or do I need to repeat anything? Let me know. If there is no doubt, then I will move to the next problem. Okay. Okay, let me go to the next problem then. So problem number 13, it says that the enzyme which catalyzes fibrinogen to fibrin conversion is number A, trypsin, number B, thrombin, number C, prothrombin, number D, pepsin. Which enzyme catalyzes the fibrinogen to fibrin conversion? The enzyme which catalyzes the fibrinogen to fibrin conversion, what will be the correct answer? Trypsin, thrombin, prothrombin, pepsin? Let me know in the chat box A, B, C or D which of the following you think is the correct answer. I will wait for a minute. The enzyme which catalyzes the fibrinogen to fibrin conversion is trypsin, thrombin, prothrombin and pepsin. Which will be the correct enzyme? The enzyme which catalyzes the fibrinogen to fibrin conversion is trypsin, thrombin, prothrombin or pepsin. Let me know A, B, C or D. I will check the chat box. Okay, let me check the chat box because we have a couple of questions left which I have to discuss. Okay, let me check if there is any answer. Okay, I got one answer which says it is number B. Let me check if number B is the correct answer or not. Okay, let me check now. Number B is correct answer or not? Only number B I have got as answer, right? Okay, now let me check it. So the correct answer will be the enzyme which catalyzes fibrinogen to fibrin conversion. It will be number B, thrombin. Okay. So basically, it is involved in our blood clot formation. So whenever we have a wound, there is a bleeding. Okay, but after some time the bleeding stops because there is a formation of the clot or by the platelet aggregation. So this is known as the blood coagulation. Okay. So what is the particular steps? So first is the clot initiation. Okay, the platelet aggregation happens, which activated the coagulation, which induces the or which activates the thrombin, and thrombin catalyzes the fibrinogen to fibrin conversion. These fibrin polymers, they form the particular clot, and the fibrinolysis happens after the retraction. When the wound heals, this retraction happens, and fibrin monopolymers means the fibrin fragment by the fibrinolysis by dissolving the particular clot. Okay. So this is first prothrombin is converted to thrombin by the prothrombin activator and calcium plus 2 ion. Then thrombin is converted to thrombin activator. Actually thrombin, it catalyzes the fibrinogen to fibrinogen monomer to fibrin conversion by in presence of calcium 2 ion, and fibrin fibers, they actually form the cross-linked fibrin fibers, and thrombin is activated fibrin stabilizing factors. It also generates, which stabilizes this cross-linked fibrin fibers. Okay. So what is the difference between thrombin and prothrombin? Basically, thrombin active enzyme that converts fibrinogen to fibrin or clot formation. Prothrombin is the inactive protein precursor of the thrombin. Prothrombin produced during the blood clotting cascade. Prothrombin present in the blood plasma waiting for activation. Thrombins require no activation, but prothrombins require an activation by other blood clotting factors like factor X. Okay. So this is the difference between thrombin and prothrombin. So is it clear or is there any doubt? So I think it is fine. If you have any doubt, you can let me know. Okay. So prothrombin, actually the precursor form of the thrombin, which is activated by the prothrombin activator, and thrombin, it catalyzes the conversion of fibrinogen to fibrin. Okay. So now I will go to the next problem. Problem number 14. Which of the following statements regarding mature prothrombin is true? Your options: number A, mature prothrombin has 10 of the glutamyl residues in the first 42 residues from amine terminus. Number B, the carboxylation process is vitamin K independent. Number C, in mature prothrombin, the glutamyl residues are modified as a gamma carboxyglutamyl residues. Number D, gamma carboxyglutamyl residues are good chelators of the magnesium plus 2 ions. Which of the following statements regarding the mature prothrombin is true? Again, I will repeat. Number A, mature prothrombin has 10 of the glutamine residues in the first 42 residues from amine terminus. Number B, the carboxylation process is vitamin K independent. Number C, in the mature prothrombin, the glutamyl residues are modified as the gamma carboxyglutamine residues. Number D, gamma carboxyglutamine residues are good chelators of the magnesium plus 2 ions. Which of the following statement is true? Since we have couple of questions left, I will be little quick. So I will wait for only a minute, then I will check the chat box. Okay. But you read the options carefully and let me know which of you think is the correct answer. Even if your answer is wrong, it's fine. Which of the following statements regarding mature prothrombin is true? Number A, mature prothrombin has 10 of the glutamyl residues in the first 42 residues from amine terminus. Number B, the carboxylation process is vitamin K independent. Number C, in mature prothrombin, glutamyl residues are modified as a gamma carboxyglutamyl residues. Number D, gamma carboxyglutamyl residues are the good chelators of the magnesium plus 2 ions. Let me check the chat box now. Okay, I got two answers, number C and number D. Let me check which of the following is the correct answer, number C or number D. So the correct answer, the statement which is true will be yes, it is number C. In mature prothrombin, the glutamyl residues are modified as gamma carboxyglutamyl residues. Okay, so these are the glutamate residues close to NH2 terminal group of the pre-prothrombin, and this is the gamma carboxyglutamyl residues. Okay, so so this gamma carboxylase is the is your enzyme, and this is a good chelator of the calcium plus 2 ions. So what the conversion of pre-prothrombin, the prothrombin is a crucial step in the synthesis of the mature functional prothrombin in the liver, which requires the vitamin K. Pre-prothrombin is an inactive precursor that undergoes post-translational modification before becoming the active coagulation factor. The conversion process location, the conversion occurs within the liver cell hepatocytes. Enzymatic activation, vitamin K is necessary cofactor for the enzyme gamma glutamyl carboxylase. The carboxylation, the enzyme modifies specific glutamic acid residues in the pre-prothrombin molecule. It adds a carboxyl group in the side chain of the glutamic acid residue, creating gamma carboxy glutamyl residues here like this. This calcium binding, these residues are essential for the mature prothrombin to bind when the calcium ions release. Once these modifications are complete, the functional prothrombin is secreted into the bloodstream. What is the significance of this gamma carboxylation? The gamma carboxylation step is critical for the proper function of the prothrombin in the blood clotting cascade. Membrane binding, the newly formed glutamate residues allow the prothrombin to bind negatively charged phospholipid surfaces such as the found on the membranes of the activated platelets. Prothrombin complex formation, binding to these membranes are essential for prothrombin to be correctly positioned for activation by the prothrombinase enzymes made of the factor XA and VA, which converts the prothrombin into the enzyme thrombin. Okay. So if there is any doubt, let me know in the chat box. I'm moving to the next question, but I will check the chat box at the end. Okay. So problem number 15, it says warfarin is a dash, fill in the blanks. Number A, vitamin C agonist. Number B, vitamin C antagonist. Number C, vitamin K agonist. And number D, vitamin K antagonist. Warfarin is a which type of molecule? Your options are number A, vitamin C agonist. Number B, vitamin C antagonist. Number C, vitamin K agonist. And number D, vitamin K antagonist. Warfarin is a which type of molecule? Vitamin C agonist, vitamin C antagonist, vitamin K agonist, or vitamin K antagonist? I will wait for a minute, then I will check the chat box. Warfarin is a which type of molecule? Warfarin is a dash. So warfarin is a which kind of molecule? Vitamin C agonist, vitamin C antagonist, vitamin K agonist, or vitamin K antagonist? Let me check the chat box. Okay, so I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. So warfarin is a Yes, number D, it is a vitamin K antagonist. What is the antagonist? What is agonist? I have already discussed in our previous session, so I am not going to repeat it. So this is warfarin, the structure of warfarin. So warfarin, the brand names also include the Coumadin and Jantoven, is a widely prescribed oral anticoagulant medication used to prevent and treat dangerous blood clots. It works by decreasing the clotting ability of the blood and also is referred as a blood thinner. Although it does not actually thin the blood. Warfarin is a vitamin K antagonist. It works by interfering the recycling of vitamin K in the liver, which is essential for the synthesis of several clotting cofactors like factors 2, 7, 9 and 10. By reducing the amount of the functional clotting factors in the blood, warfarin prolongs the time it takes for the blood to clot. Warfarin is used to prevent and treat various thromboemic conditions, including venous thromboembolism, the blood clots that form in vein, such as deep vein thrombosis, most commonly in legs, pulmonary embolism, a clot which is traveled to the lung, often originated from a DVT, arterial fibrillation, irregular heart rhythm that can cause blood to pool and form clots in the heart, increasing the risk of the stroke. Heart valve replacement to prevent the clot from forming or on mechanical or bioprosthetic heart valves following a heart attack to reduce the risks of the recurrent heart attacks and other thrombotic events. Okay, so these are the uses of the warfarin. So remember, warfarin is a vitamin K antagonist, and this is the structure of the warfarin. Okay, this is a very simple or easy structure. You can easily remember it. Now I will go to the next problem, problem number 16, which says which of the following statements is false regarding the methotrexate. Your options are: number A, it is a potent anti-cancer drug. Number B, it works by catalyzing the dihydrofolate reductase. Number C, the interaction is non-covalent. And number D, the crystal structure is known. Which of the following statements is false regarding the methotrexate? Number A, it is a potent anti-cancer drug. Number B, it works by catalyzing the dihydrofolate reductase. Number C, the interaction is non-covalent. Number D, its crystal structure is known. Which of the following statements is false regarding methotrexate? Again, it is a true-false kind of question, so read the options carefully. I will wait for a minute or two, then I will check the chat box. Okay. Which of the following statements is false regarding methotrexate? Let me know. Number A, it's a potent anti-cancer drug. Number B, it works by catalyzing dihydrofolate reductase. Number C, the interaction is non-covalent. And number D, the crystal structure is known. Which of the following statements is false regarding methotrexate? Let me check the chat box now. Let me see if there is any answer for problem number 16. Okay, I got one answer which says it is number C. Let me check if number C is false or not. So, the statement which is false regarding methotrexate will be, it is number B. It works by catalyzing the dihydrofolate reductase. Okay, so this is the structure of methotrexate. So actually, methotrexate is a dihydrofolate reductase. Okay, so this is the what happens in this case of the folic acid metabolism. So dihydrofolate reductase is an enzyme which is crucial for maintaining levels of the enzyme cofactor tetrahydrofolate. Without this cofactor, the synthesis of DNA building block DTMP would grind to a halt. This is in turn would slow down the DNA synthesis and cell division. The enzyme catalyzes the reduction of the vitamin folic acid the 2 FH4 in two step where dihydrofolate once formed FH4 is picked by single carbon unit to form N5N10 methylene FH4, which acts as a source of one carbon unit in various biosynthetic pathway, including the methylation of the dihydroxyurine monophosphate or DUMP to form the DTMP. This I have already discussed, right? When I was discussing the thymidylase synthase, you can see up to this we have discussed, right? The FH4 conversion from folic acid by this enzyme we have discussed. After that, this FH4 is converted to N5N10 methylene H4, which is the source of the methyl group, which is added to the DUMP to DTMP by the thymidylase synthesis. Okay, so this is the aftermath which happens after the formation of the tetrahydrofolate. The N5N10 methylene FH4 is converted back to FH2 in the process. Okay, and the dihydrofolate reductase is a vital restoring of the F5F methylene FH4 for further reaction. Methotrexate is one of the most widely used antimetabolites in cancer chemotherapy. It is very similar to in the structure of the natural folates, differing only in additional amino and methyl group. It has a stronger binding affinity for the enzyme owing an additional hydrogen bond or ionic bond which is not present in FH2 binds. As a result, methotrexate prevents the binding of FH2 in conversion to the N5N10 methylene FH4. Depletion of the cofactor has the greatest effect on the enzyme thymidylate synthase or TS, resulting in lowered synthesis of DTMP. Methotrexate tends to accumulate in cells as a result of the polyglutamylation. This is an enzyme catalyzed in the process which involves the addition of the glutamate groups to the glutamate moiety already present in the molecule. This also happens to the natural folate. The reaction serves to increase the charge and the size of the folate such that they are trapped within the cell. Pemetrexate and pralatrexate are the related drugs that were approved in 2004 and 9 respectively. Okay, so this is all about the methotrexate. I will go to the next problem. The hydrogens which are most likely to get exchanged with the deuterium when the following molecule is dissolved in D2O. This molecule, your options are number A HA only, number B HB and HD, number C HB and HC, and number D HD only. Which hydrogens are most likely to get exchanged with deuterium when the following molecule is dissolved in D2O? We don't have much time, so I will wait only for a minute. Let me know A, B, C or D. Which of the following is the correct answer of problem number 17? Which are the hydrogens that are most likely to get exchanged with deuterium when the following molecule is dissolved in D2O? HA only, HB or HD, HB or HC or HD only? What will be the correct answer for problem number 17? Which hydrogens are most likely to get exchanged when this molecule is dissolved in D2O? The options are HA only, option B HB and HD, option C HB and HC, and option D is HD only. The hydrogens that are most likely to get exchanged when the deuterium, when the following molecule is dissolved in D2O. Let me check the chat box. Okay, I got one answer, which is number C. Let me check if number C is the correct answer or not for problem number 17. So the correct answer, yes, it is number C, HB and HC. This HB and HC are the most acidic protons which are most likely to get exchanged when this molecule is dissolved in D2O. So deuterium exchange peaks that are due to the OH and NH protons can be identified by the exchange of deuterium. Deuterium is not observed in the proton NMR spectra. Shaking a sample containing exchangeable protons with D2O causes the exchangeable proton to be replaced by deuterium. As a result, the signal for the exchangeable proton disappears from the spectrum. Okay, so these are the reactions. So this is the hydrogen deuterium exchange in NMR spectroscopy. This is the OH peak is present here, but when the compound is dissolved in D2O, the exchangeable protons are replaced by deuterium, and there is no OH peak is present because this peak disappears since deuterium is NMR inactive at the proton's frequency. Okay. So that is how it happens. So if you have any doubt or any query regarding any question, please let me know in the chat box if I need to repeat anything. Let me complete the questions, and then if you have any doubt, I will happily answer those. Okay. Let me go to the next problem. Problem number 18. Which of the following reactions is catalyzed by vitamin B12 coenzyme? Your options are number A, homocysteine to methionine conversion, number B, methyl manonyl CoA to succinyl CoA conversion, number C, none, and number D, both. Which of the following reactions is catalyzed by vitamin B12 coenzyme? Number A, homocysteine to methionine conversion. Number B, methyl malonyl CoA to succinyl CoA conversion. Number C, none. Number D, both. Which of the following reactions is catalyzed by vitamin B12 coenzyme? Number A, homocysteine to methionine conversion. Number B, methyl malonyl CoA to succinyl CoA conversion. Number C, none. Number D, both. Let me know. I will wait for a minute. Which reaction is catalyzed by vitamin B12 coenzyme? Which of the following reactions is catalyzed by vitamin B12 coenzyme? Let me check the chat box. Okay, number D is the given answer. Let me check if number D is the correct answer or not. So the correct answer will be, it is number D. Yes, both. Both the reactions. So vitamin B12 or cobalamin is a cofactor in the two critical metabolic reaction within the human body, the conversion of the homocysteine to methionine and the conversion of the methyl manonyl CoA to succinyl CoA. These reactions are essential for the DNA synthesis, cell growth, energy production, and health of the nervous system. Homocysteine to methionine conversion. The methionine synthesis is the enzyme that uses methyl cobalamin as a cofactor to catalyze this reaction function. It transfers the methyl group from the 5-methyl tetrahydrofolate, a form of the folate, to homocysteine, producing methionine and tetrahydrofolate. Importance: methionine is essential amino acid and precursor of the S-adenosyl methionine or SAM, the body's primary methyl donor. The regeneration of the THF is crucial for the DNA synthesis. Consequences of the deficiency: without enough methyl the homocysteine levels rise and the folate becomes trapped in a form that cannot be used for DNA synthesis. This leads to the megaloblastic anemia, which is characterized by large and immature red blood cells. And methyl manonyl CoA to succinyl CoA. Methyl manonyl CoA mutates as ADOCBL as a cofactor for this reaction. Okay. And function: this reaction is a vital step for the catabolism or breakdown of certain amino acid, old chain fatty acid, and cholesterol. Importance: the product succinyl CoA is the key intermediate for the citric acid cycle, which is central cellular energy production. Consequences: on deficiency: impaired MUT activity causes the buildup of the methyl mannonic acid. Elevated MMA is toxic and is thought to contribute in the neurological damage often seen in the severe B12 deficiency. Okay. Now problem number 19, it says glycerol 2,3-hydroxypropanoic acid conversion is converted by which enzyme? Number A, glycerol hydratase and alcohol dehydrogenase. Number B, glycerol dehydratase and alcohol dehydrogenase. Number C, glycerol dehydratase and aldehyde dehydrogenase. And number D, alcohol dehydrogenase and aldehyde dehydrogenase. Glycerol to 3-hydroxypropanoic acid conversion is catalyzed by which enzymes? Again, I will wait for only a minute. Your options are number A, glycerol dehydratase and alcohol dehydrogenase. Number B, glycerol dehydratase and alcohol dehydrogenase. Number C, glycerol dehydratase and aldehyde dehydrogenase. Number D, alcohol dehydrogenase and aldehyde dehydrogenase. Glycerol to dihydroxypropanoic acid conversion is catalyzed by which enzyme? Let me know A, B, C or D, which of the following will be the correct answer. Let me check the chat box because we have another question left which we have to discuss. Okay, let me check if there is any answer. Okay, I got one answer saying it is number B. Let me check if number B is the correct answer or not. So number C is the correct answer actually. Glycerol dehydratase and aldehyde dehydrogenase are the correct set of enzymes. So as you can see, there is a conversion of the glycerol to 3-hydroxypropanol. Okay, so the hydroxypropanol is the aldehyde form, as you can see, and the enzyme which is responsible is the glycerol dehydratase. As you can see here, it is given. Let me highlight it for you. Okay, so glycerol dehydratase converts glycerol to 3-hydroxypropanol. 3-hydroxypropanol is converted to 3-hydroxypropanoic acid by the enzyme aldehyde dehydrogenase. Okay, and so the conversion of glycerol to 3-hydroxypropanoic acid is catalyzed by glycerol dehydratase and aldehyde dehydratase. And these are the intermediate is the 3-hydroxypropanol is also from 3-hydroxypropanol is converted to 3-hydroxypropanol CoA by the propanaldehyde dehydrogenase, which is converted to 3-hydroxypropanol phosphate and 3-hydroxypropanoic acid. Okay, so this is 3-hydroxypropanoic acids are also can be synthesized in this pathway. Okay, so the correct answer was number C. Now, last question of today's session. The side product formed at the time of the conversion of the serine to glycine through the serine hydroxymethyl transferase is number A, benzaldehyde, number B, formaldehyde, number C, acetaldehyde, and number D, acetone. Which is the side product which is formed at the time of the conversion of the serine to glycine through the serine hydroxymethyl transferase? The side product formed at the time of the conversion to serine to glycine through the serine hydroxymethyl transferase. Let me check the chat box. Let me check if there is any. This is the last question. So let me check the chat box if there is any correct answer. I got one answer which is saying it is number B. Now let me check if number B is the correct answer or not. So the correct answer will be yes, it is number B. Formaldehyde is the side product formed at the time of the conversion of serine to glycine through the serine hydroxymethyl transferase. So this is the glycine cleavage system. Okay, so serine hydroxymethyl transferase, another pyridoxal phosphate dependent enzyme. So this is important. That's this serine hydroxylase is a pyridoxal phosphate dependent enzyme that catalyzes the interconversion of the amino acid serine to serine with glycine and formaldehyde. And the proposed so this is the thing. So this do not need to answer. So this is the serine hydroxymethyl thing. This is how the conversion happens, and this is the glycine cleavage system. Okay, so you do not need to memorize all these things. Just look at the conversions how it happens. Okay, but you do not need to memorize these things. Okay. So this was the last question of today's session. So today what we did, we have discussed regarding the various cofactors and coenzymes in various enzymatic reactions and various conversions. Mostly we have discussed how folate system had folic acid metabolism happens and what is the roles of various cofactors and coenzymes. We discussed how thymidylate synthesis is why it is important, how it is connected with the folic acid metabolism. We discussed about the blood clotting and on these things, and also we have discussed about miscellaneous enzymes. Okay, so this was the last question of today's session. So I will share my email id. So this is my email id. If you have a, a second. So this is my email id. If you have any doubt or any query, please let me know through the email. If there is any doubt in today's session, if yes, you can let me know. This presentation and class recording I will be sharing by today. So you can go through it later also. And if you have any doubt, you can let me know via email. Okay. So this was the week 8 live session for this NPTEL course of organic chemistry and biology and drug development. We will meet on next Friday. I think the next Friday will be 19th September, and for our week 9 live session. Meanwhile, if you have any doubt regarding any course content which we have covered till now, please let me know via email. Thank you all for joining. I will close the session for today.